Yeah, thank you. Good evening, friends. This is Dr. I.R. Rajkumar welcoming you yet again on behalf of IPS Vadodara and Bombay Psychiatric Society for a session on role of gut microbiota in psychiatric illnesses. Dr. Vihang Vahia is the chairman for this evening. I don't think Dr. Vihang Vahia needs any introduction to any of us. A very prolific speaker and academician at Bombay, who we have been knowing for the last 50 years. Dr. Vihan, here on it is you managing the show.
Am I audible now? I became mute.
Yes, you are.
Thank you. Good evening, colleagues. It's indeed a privilege to chair this session on gut-brain axis. For an Indian audience, the concept of gut-brain axis is almost 10,000 years old. Indian literature and traditions always talk of gut health for mental health. We have always had traditional diet in different parts of the country, which is modified depending upon the physical health and the mental health. And our ancestors are traditionally people spoke of food which is hot and food which is cooling. That is food which aggravates mental agony and food which facilitates mental agony. On psychiatric literature, the gut-brain axis was earlier described by the gastroenterologists. And in 2013 was the first time that I read an article on FMPP, fermented milk product with probiotic, and its effect on anxiety and neuromodulation. Interestingly, FMPP in terms of Indian dietetics would mean lassi. Lassi made with cultured yogurt or dahi. We can't have tons of, from 5-6 liters of lassi a day to keep our mind cool. And that's how came up the concept of use of probiotics in multiple psychiatric conditions, particularly anxiety and anxiety-related disorders. There is so much literature that's coming up on the gut-brain axis that today, even a new entrant into medical field, like a second MBA student, would be able to recite or tell us about his information or her information on gut-brain axis. We need to go a little beyond second MBBS knowledge. And to do that, we have Dr. Manish Roshan Thakur, who's going to tell us a lot more about the gut-brain axis. For those of us who don't know him, he did his psychiatry, MD psychiatry from AIMS, New Delhi. He was formerly a consultant at IIT Delhi and is currently practicing psychiatry at the, and he presented he got his award actually as a best paper in social psychiatry. It's interesting that a probably biological psychiatrist got his award in a social psychiatry at the National Conference of the Indian Association of Social Psychiatry in 2023 at Raipur. He has been a student of neuroscience and is struggling to find the meaning of brain. Indeed, Dr. Manish, a tall order. He runs an educational platform for the postgraduate residents called the Psych Reads, where active, authentic, and scientific learning is conducted, I guess, personally by Dr. Manish. He has, in his short career, published 10 papers in national and international journals and is interested in biological psychiatry, psychopharmacology, genetics, and neurodevelopmental disorders. He's going to talk to us today on neuromodulation as well, I guess. Dr. Manish, Over to you, and we look forward to a real learning experience in this emerging field of gut brain access and mental health. Over to you, Dr. Manish.
Uh, Thank you, sir. First of all, good evening to everyone. It's indeed a pleasure to be here. And let's start with the topic. The topic for today's discussion is role of gut microbiota in psychiatric illness. Now, yeah. So before starting, see, all animals, including the humans, evolved an innate association with microbial communities comprising of the bacteria, archaea, fungi, and viruses. These collection of microorganisms, termed as microbiota, inhibit inhabit nearly every environmentally exposed body surface, which in the community, it might be in the GIT or in the oral mucosa, representing the greatest diversity and absolute abundance of microorganisms in human body. Rather than being passive passengers in the human body, they are considered with whatever research that we have uh, that has been done and the data that has come up, it has revealed that it is of utmost crucial importance that it is able to modulate the functions of the brain. Gut bacterial communities are dynamic that can change both in comparison and activity throughout our lives and in response to host factors such as the age as well as the genetics and to the changing environment. Bacteria found in gut microbiota is equal to the number of the human cells in the body. And the genetic repertoire, if you look, it is around 232 million genes, which greatly expand the metabolic potential of the humans. So with this brief introduction, we'll start with the mammalian nervous system, which is organized into two subdivisions, the central nervous system and the peripheral nervous system. And the CNS includes the brain and the spinal cord. The PNS includes the ganglia nerve branches. Autonomic nervous system regulates essential visceral processes, the sympathetic and the parasympathetic systems. And the enteric nervous system is a branch of autonomic nervous system, which is crucial for maintaining a stable gut health. Now, the communication between the CNS and the ENS travels to a different signaling pathway, which might include a direct neuronal pathway or by other means like the endocrine pathway. Now, the gut microbiota brain axis. See, the gut microbiota, which is present in the GI tract, there is a bidirectional communication that is between, just a minute. There is a bidirectional communication between the gut microbiota and CNS mediated by several direct and indirect pathway. Now, the route of communication, it involves ANS or the autonomic nervous system, the ENS, neuroendocrine system, the HPA axis, that is the hypothalamus, pituitary axis, and immune system and other metabolic pathway. Now, before starting with microbiome and microbiota, let's look at what are they and what do they mean. So microbiome is collectively refers to the genetic material of the entire microbial community residing on and in the human body, which encompasses bacteria, eukaryote viruses, fungi, protozoa, archaea, and bacteriophage. And microbiota refers to the entire microbial community residing on and in human body, which encompasses the bacteria, eukaryote viruses, fungi, protozoa, archaea, and bacteriophage. With this basics, let's start with the normal gut microbiome. So the gut microbiota per se acts as a filter and a biological rheostat for sensing, modifying, and tuning vast amount of chemical signals from the environment that then circulate throughout the body. As such, the gut bacterial communities lies at the intersection of the host and the environment and may directly influence the human health. Now, the gut microbiota brain axis, it refers to the network of connections involving multiple biological systems and allow bidirectional communication between the gut, bacteria, and the brain. So in the screen, we can see that what are the various common micro uh, biota that is present, be it in the stomach, small intestine, ileum, or the large intestine. Okay. Now, specifically, if you look up and look into what are the major 7 divisions, so this they have given from the top to the bottom. So Firmicutes, bacteroidetes, proteobacteria, fusobacteria. Okay, these are in the sequence, and the role has been mentioned, like be it protective, metabolic, or the structural role. Now, the gut biota within the gut, the microbiota can produce neuroactive compounds either in form of neurotransmitters or amino acids or microbial metabolites, like for example GABA, norepinephrine, dopamine, or serotonin, or amino acid like tyramine or and tryptophan, microbial metabolites like short chain fatty acid and 4-ethyl phenyl sulfate. These metabolites then enter the portal system, and through the portal system they communicate to the local neuronal cells of the they communicate to the other parts of the body, which might include the host immune system, the local neuronal cells of the enteric nervous system and the vagus nerve, which ultimately influence the metabolism and thus our brain. Now, looking into the gut barrier integrity. So, if you look at the histology of the gut, so the microbiota influence the gut barrier integrity that controls the passage of signaling molecules from the gut lumen to the lamina propria. So, this part is the lumen, this part is the lumen, and from here we have the epithelial cells, and the basal part here is the lamina propria. And the lamina propria contains immune cells and the nerve endings of the entering nervous system, the neurons, and also the route to the portal circulation begins from here. If you just look into the epithelial cells, here there are a multiple uh, proteins which are present in junction of the two cells, be it desmosome that is represented through through the blue color, be it the gap junctions, the occludins that is represented through the black color. So these are some of the important proteins which helps in maintaining the integrity of the gut layer. Now, a step ahead, let's look at the tight junction protein functioning, which is present between two of the epithelial cells. So the TJ protein or the tight junction protein regulate the rate of paracellular transport. That is between the cell, if we look, the cellular transport from the basal side to the apical side and through in between the two cells, the transport that is called as the paracellular transport is being regulated by the TJ proteins, and it includes the transport of nutrients between neighboring epithelial cells. These TJ proteins act as a sensor of environment condition that dynamically regulate the paracellular transport of solute. And dysregulation of TJ protein can lead to excessive permeability of the intestinal barrier. Now, just an example of how the TJ proteins are getting affected is when we look into the pathogenic strains of Escherichia coli, that is O157H7 strain, which produce toxins, it suppresses the protein synthesis of tight junctions and ultimately lead to HUS. And the non-pathogenic E. coli can decrease the expression of TJ proteins, and it increases the production of the toxin STX2A by the non-pathogenic strain E, of E. coli. Now, if you just look at the probiotics and the TJ protein, the relationship between the two, probiotics such as the lactobacillus and bifidobacterium species, they contribute to the integrity of the intestinal barrier. Lactobacillus reuteri increases the expression of TJ protein and supporting the integrity of the intestinal wall. Also, bifidobacterium and lactobacillus acidophilus prevents dysregulation of occludin and claudin 1 level in colon cancer cell line. So these are, if you look, the claudin and occludin has been shown here on the right side of the picture in blue color. So these are those proteins which is being regulated by lactobacillus and bifidobacterium. Now, when we give antibiotics to treat these kind of infections, the gut infection, antibiotics used to treat bacterial infection may adversely affect the gut microbiota, causing an imbalance between the specific bacterial group, and it triggers dysbacteriosis. Okay, dysbacteriosis contributes to increased intestinal permeability. Now, if you just have a look at the various bacterial strain and how they affect the expression of these tight junction proteins, whether they are increased or decreased. So this is a list which provides the relationship between the lactobacillus species and bifidobacterium species, which shows that some of these cells, so some of these bacterial strains, they actually lead to an increased production uh, of the zona occludens or occludin or claudin, which helps in maintaining the gut barrier integrity. If they are lost, they will definitely lead to decrease in production of tight junction protein, and ultimately there will be a breach in the barrier, or there will be loss in the barrier integrity also, which will ultimately affect the paracellular transport. If these bacteria are being lost because of the antibiotic use, or because of the infection, or because of autoimmune conditions, or any of the reasons, there will be a loss to the barrier proteins. Now, apart from these barrier proteins, on top of that, we have something that is called as the mucus layer. And the mucin protein, which is comprising of 95% water, it assembles into long polymers that hold the bacteria and keep a layer of bacteria on the top of the cells. Now, the gut microbiota can modulate the mucus proteins, the properties which affects the glycan surface, uh, the glycan profile through glycosyl transferases. Now, bacteroids species, they activate alpha-1,2 fucosyl transferase, which creates a selective habitat for the gut microbial community. Now, gut microbiota may degrade and metabolize the mucin glycans, and the mucus becomes less viscous. Hence, it becomes more permeable to toxin and microbes. Thus, alteration in the mucus layer can affect the composition and biodistribution of the gut microbiota. So, two of the mechanisms we have seen. The first one was the loss of integrity of the um, of the epithelial cells that was caused by alteration in the tight junction proteins or other proteins like zona occludens, claudins, and occludins. And the second one is the alteration in the property of the mucus layer. Now, coming to the other mechanisms like the oxidative stress. So oxidative stress, and it usually represents an imbalance between the generation and elimination of the ROS, that is the oxidative species, which can lead to an intestinal barrier dysfunction. So whenever there is rise in the oxidant or the oxidative symptom, that will lead to an oxidative stress, which will ultimately cause damage to the cell. Okay, the damage to the cell is not only limited to the structural damage. There is a damage which is being caused to the DNA, ultimately affecting the metabolism, that is lipid peroxidation and protein carbonylation. Mitochondrial electrocyte protein, the chain converts less than 10% of consumed oxygen to the ROS, causing basal steady state oxidative stress. Now, when there is increased ROS level, it can lead to low, intermediate, and high intensity oxidative stress. Probiotics like, for example, lactobacillus and bifidobacterium, they can neutralize these ROS and can increase the antioxidant level. Lactobacillus plantarum, it exhibits a strong antioxidant effect, and it is possibly lead to the secretion of the L-ascorbic acid. So a balance is being maintained between the oxidative system and the antioxidant system, so which keeps the cell cycle in check and also will help in maintaining the longevity of the cells. Now, apart from whatever mechanisms we have discussed till now regarding how the barrier integrity is being maintained and how these set of microbiota is able to maintain a healthy environment in the gut, there are some of the other proposed mechanisms by which it regulates the nervous system and which ultimately leads to the behavioral changes. Like, for example, if you look for the first of the gut microorganism, that is the Lactobacillus reuteri, it leads to increased production of oxytocin, and ultimately it regulates the neuronal plasticity, helping in regulating the social behavior. Also, if you look through the other organism, the Lactobacillus rhamnosus, it increases the flow of GABA, and it decreases the stress responsiveness, decreases anxiety, and also depressive like behavior. And the vagal tone in the mesenteric nerve firing is also being regulated by this. Bifidobacterium, on the other hand, will lead to the production of the BDNF, which also decreases the excitability of the enteric nervous system and also decreases anxiety and depressive like state. Bacteroides fragilis, it decreases the production of the 4-EPS, which further decreases the anxiety like behavior. Subsequently, the short chain fatty acid producing bacteria, it leads to a increased production of short chain uh, uh, fatty acids, which decreases the stress, decreases anxiety and depressive like behavior. So these are some of the proposed mechanisms that, apart from maintaining the integrity of the gut barrier and the mucus layer, as well as by maintaining an antioxidant environment, these are some of the proposed mechanism by which these bacteria are able to act. Now, see, let's look into the impact of the on the neurotransmission. So, apart from all those things that we have seen in a while, like we have seen that a number of processes is being regulated. It is also having an impact on the neurotransmitters. So, several neurotransmitters have been isolated from the microbial species that are known to occur in the human gut. And if we just look through that through this gray box, so every species is able to modulate a set of neurotransmitter that has been shown over here in the lower gray box. And if you look through that, the microbial production of neurotransmitters, it represents a potential mechanism to directly influence the brain and the behavior. Although this route is limited, although in reality this route is limited because most of the neurotransmitters, including the serotonin, dopamine, and GABA, they typically they cannot breach the protective blood-brain barrier. So there is a distinction between the production of the neurotransmitters in the other section of the body and inside the brain. And thus, there are alternate modes of action that is possible by these microorganism derived neurotransmitter that affect the brain. It can be through the vagus nerve and through its afferent neurons. Also, if you look, there are other mechanisms like, for example, the precursors of the neurotransmitters. They can cross the blood-brain barrier and then they get converted into the active neurotransmitters. For example, if you look, gut bacteria can influence serotonergic signaling in the CNS, as tryptophan concentration in the blood plasma has been shown to correlate with the blood serotonin level. So that might be the possible ways or the mechanism by which the neurotransmitters, which are being the production of which is being regulated by these subset of bacteria, might alter the neurotransmission. So whatever we have looked till now, be it the drugs or the antibiotics, the diet, the environment, the pathogens, or the genetic susceptibility, they, if at all present, they lead to the alteration of microbiota, which will ultimately cause damage to the epithelial layers. And whenever there is damage to the epithelial layer, there will be a leakage of endotoxins. Further, there will be alteration in the paracellular transport. All of them will lead to two subset of two big response. One is the immune response, which leads to the neuroinflammation or inflammatory condition in the body, and the other will be the toxemia or the endotoxemia, which will further lead to a low grade systematic inflammation. And these might be the cause, that could be the responsible for development of the subset of illnesses that we are going to discuss today. Now, today we'll be looking and we'll be restricting our discussion to these subset of disorders. So first of all, let's look into the bipolar disorder, followed by depression, schizophrenia, autism, and neurodegenerative disorders. So pretty much the mechanism or the pathophysiology will remain the same, but there will be some kind of alteration that we are going to see. So first, starting with gut microbiota and mood disorder. So there is a connection between the brain and the gut microbiota in stress, and that is bidirectional, as chronic stress is associated with lasting alteration in the composition and functioning of gut microbiota. Now, with uh, the usual population of the gut microbiota, there is a environment that is being created which is considered to be normal, and thus there is a homeostasis which is maintained in the region of the gut. But whenever there is chronic stress, there will be alteration in the composition and functioning of the gut microbiota, which will lead to the setup of a new equilibrium that will be called as the allostasis. Now, the allostasis, it involves the dynamic regulation of the body's stress response system that will include the neuroendocrine signaling through the hypothalamic pituitary axis, which regulates the production of glucocorticoids. If we look at the gut microbiota, they try to restore this allostasis so that the basal metabolism and the usual environment that used to be there in the gut, that might be restored. So while during stress, administration of lactobacillus species, they normalize the level of glucocorticoids following the early life stress. Uh, Rhamnosus species of lactobacillus, they elevate the anxiety-like behavior. This has been seen in rodents. Bifidobacterium, they have shown to ameliorate stress-induced behavioral alterations in rodent models. Gut microbiota can also affect the stress response pathway in the brain. Now, just having a look at this, so if we just look at the red boxes over here that has been shaded in the lower segment of the picture, then when we look through the individuals or when we look through the microbiota ecosystem of the bipolar disorder, as shown, the bipolar disorder patients are characterized by representing a reduced microbial diversity and changes in the relative abundance of the several taxa. Okay, so there will be an alteration in the usual gut microbiome, and also some subset of gut microbiota will be definitely being increased and the other subset will be reduced. Now, uh, now the abundance of several taxa which will be correlated with the pathogenesis of the complex disorder has been shown in the lower segments. Now, besides specific disturbance according to the type and clinical manifestation, although further distinction between type 1, type 2, and the mixed episodes and the correlation with that of gut microbiota still that is coming up. Yet, some of the findings that is there for type 1 and type 2 bipolar disorder has been shown over here. Like in type 1, there is an increase in IgM response to Moragnella, and in type 2, there is increase in the population of Collinsella. So, but the distinction between type 1, type 2, and mixed episode based on the gut microbiome, still the studies are going on. But compelling evidence is also supporting the notion that gut microbiota can present some similarities and dissimilarities across the psychiatric disorder. And this might be one of the potential biomarkers which might be involved in uh, drug the early detection of the bipolar disorder and also in the treatment modality. Now, if we just look at this, so factor involved, usually if you see the gut microbiota dysbiosis will happen, which will ultimately lead to the leaky gut, or there is loss of the integrity of the gut barrier. There will be bacterial translocation, which will lead to endotoxemia. Now, with endotoxemia and with the leaky gut, there is bacterial translocation which is happening, which leads to intestinal inflammation, which gradually leads to systemic inflammation. Now, three of the layers, the mucus, uh, the uh, the epithelial layer, they are being damaged. Apart from this, there is a change in the oxidative stress level. So there will be more production of reactive oxidative species, and ultimately it will have an effect in the brain that will lead to a state of neuroinflammation. Now, with this state of neuroinflammation, what will happen is there will be a damage to the oligodendrocytes and changes in the white matter, which will be seen in this disorder. Also, if we look, there will be a decrease in production of BDNF, which is as seen in the patients of mania and depression. So here is one of the correlates by which we can like one of the possible mechanism which is leading to this particular disorder, which is being caused by the gut microbiota dysbiosis. Now, looking for the other disorder, let's move on to depression. If we look at into the depression, now here also there is a like it has been shown in the gray and also in the brown box. There is a imbalance between the gut microbiome, and some subsets of gut microbiota is increased in the state of depression, and there is reduction in other set another in the other subset. Now, all of these will lead to the similar set of equation, that there will be a leaky gut, there will be an increased tendency to develop a state of inflammation, which will ultimately lead to changes in the brain because of the neuroinflammation. Also, there will be an activation of the HPA axis, and then there will be a state in which more of the glucocorticoids will be there. Now, similar subset of changes will be also noticed in depression, as we have seen in mania. But here, let's look at this. So, uh, if we just look at the lower section of the picture, so the gut microbiota and its metabolites have translocated through the protective layer, that is the epithelial layer, and they are leading to systemic inflammation. Also, whenever the systemic inflammation is taking place, there is a release of these various inflammatory molecules, which will ultimately lead to damage to the blood-brain barrier or will activate the hypothalamic pituitary axis. Now, the state of inflammation will be carried forward to the above portion that is there in the brain. And this is how there will be a dysregulation in the homeostasis. Now, whenever we treat patients with depression, antidepressants are being used, and this is one of the chart which provides a list of antidepressants and also how they are affecting the uh, gut microbiome. Now, here in this chart, we can see there is an effect on the alpha diversity written and also an effect on the beta diversity that has been written. Now, if we look into the alpha diversity reflects to the diversity of the species within a single sample. While the beta diversity, if we look, it compares the diversity between different sample. So alpha diversity usually focus on within sample richness and evenness, and it describes the number and the relative abundance of different uh, microbial species within that sample. Whereas the beta, conversely, it examines the differences in the species composition between different samples and indicating how dissimilar the communities are from each other. So if you look, the paroxetine and escitalopram, fluoxetine, all of which are showing a significant effect on the beta diversity. And a subset of taxa, a set of gut microbiome, gut microbiota will be reduced and other will be increased. Now, there are some of the involved metabolites that might be responsible, but if you just look at this, paroxetine, citalopram, fluoxetine is having an effect which is significantly noticed in the beta diversity that is between the sample. Also in the SNRI group, venlafaxine is also having a significant role in causing a decrease in the subset of microbiota. Now, in the TCA group, amitriptyline and desimipramin is also showing that kind of significant effect on the beta diversity. For the other subset, the data, however, is not available. Okay, so imipramine, clomipramine, phenelzine, the data per se is not there, but we can see that whatever drug that we are using, be it paroxetine, citalopram, fluoxetine, venlafaxine, amitriptyline, and desimipramin, all are having a significant effect on the beta diversity. That is, it affects the gut microbiota. Now, coming to schizophrenia. Now, see, again, we'll be keeping the pathophysiological mechanisms here, which is almost the same, but there will be some alteration that will be seen. So intestinal permeability, as we have discussed, how it is being increased, that will be there noted, and ultimately it will lead to a systematic low level inflammation, that is through the immune system involvement, and there will be a pro-inflammatory cytokine. Now, there will be modulation of tryptophan kynurenine metabolism. Ultimately, there will be altered amino acid and lipid metabolism, which leads to the disruption in the glutamine GABA cycle, reducing the brain glutamate. Long-term continuation of such a state will lead to alteration in the brain structure. Here also, most of the studies, it says that no changes in the alpha diversity is there, but there are changes in the beta diversity. That is a consistent finding in patients with schizophrenia. If we look into the first episode, psychosis, here also a link between the gut microbiome and changes in the brain structure, that is the gray matter volume, has been found. And there are also changes, but here the relative reduction in alpha diversity, that is similar sample species, in the similar sample, they have shown a reduction. And there are also certain changes which are also observed in the patients who are having the first episode of psychosis. Now, if we look through this, that some of the gut microbiota is associated with a subset of symptom, be it negative symptom, positive symptom, or global functioning of the patient or symptom severity. Some of the patients, like some of the subsets of the gut microbiota will definitely have an impact on these set of disorders. These set of symptoms. So if we look through the first one, that is the increased symptom severity, which is associated with lactobacillus, a decrease in the lactobacillus subset will lead to increased symptom severity, and lacnose spiraceae bacteroides species are also associated with this. And increased negative symptom is associated with these two species. Again, a lack of lactobacillus in the gut will lead to increased positive symptom, and decreased global functioning is associated with these three uh, subset of gut microbiota. Remember what we have just discussed, that such a kind of distinction is there in schizophrenia, but for bipolar disorder, the type 1, type 2, and the mixed episode, such kind of distinction is still not present. Now, looking into this, this is again a patient with schizophrenia, and the patient is taking antipsychotics, and what is happening is like there is a gut dysbiosis which is happening with similar subset of physiological mechanism. There is activation of the vagus nerve and also there will be other mechanisms like endocrine, and the behavioral responses are initiated. There will be changes in the level of glutamate, glutamine, dopamine, GABA, and the serotonin level. The kynurenine pathway of tryptophan metabolism will be altered. There is a state of uh, low level systematic inflammation ultimately reaching the brain, which leads to a state of neuroinflammation. And ultimately, the patients will present with schizophrenia symptoms, be it positive, negative, cognitive impairment. And there might be associated depression and anxiety like feature, which is one of the symptom of schizophrenia. Now, apart from this, if we just look not only the symptoms of schizophrenia, but also the metabolic alteration that is happening in the patients. So these might also be associated with the alteration in the gut microbiota. Now, let's move on to autism. So the idea of possible involvement of gut microbiota in autism spectrum disorder was first postulated in 1998 by Bolte, and he speculated that Clostridium tetani could induce autism. In 2001, Sandler et al. Conducted a pilot study, and he reported a temporary improvement in both the behavioral and GI symptom in autistic children after 6 weeks of treatment with oral vancomycin. There was the gradual regression of the symptomatology following the discontinuation of the treatment uh, was explained by the later. So what was happening was like it was explained by the later germination of the Clostridium spores, which were resistant to antibiotics. So there was an effect that was being noted by oral vancomycin. However, because of the resistance to the antibiotic, there was further symptomatology which was found. However, these early findings, it coupled with the frequent occurrence of the GI symptom in patients with autism spectrum disorder, and that has led to investigate the composition of gut microbiota in these subset of patients. Now, here is one of the reviews which looked into what are the various changes in the gut microbiota as far as these patients are concerned. So firmicutes and uh, the uh, the formicutes and the bacteroidetes, the actinobacteria have shown considerable enrichment in children with autism spectrum disorder by mental regression and no mental regression. So there is a kind of difference between the two. And Clostridium, they have been found to be increased in patients with autism when compared to the control. Also, there is a rise in propionic acid, which is correlated with the neurotoxic effect. Clostridium perfringens have significantly increased in the gut of autism spectrum disorder compared to the control group. And the highest level of ASD subjects or the patients with GI symptoms, they have the increased population of Clostridium perfringens. Other subsets are also reported as mucosa-associated clostritis in ASD children with GI disorder. Now, Des salvo vibrio, a type of bacteria which potentially contribute to GI complications, was also observed in autism spectrum disorder. Bacteroides species have found to be increased or decreased. So there is, like in some of the studies, there are increase in the population of this subset. Other studies have shown that there is a decrease. Now, abundance of the butyric acid, which is a short chain fatty acid, is known to for its anti-inflammatory and protective properties. However, because of the subset or alteration in the gut microbiota, the butyric acid here in autism has shown to be reduced. And further decreased level of bifidobacterium has been reported in autism spectrum disorder, and bifidobacterium, which is actually having a protective role in this subset of patients, have shown to the population has shown a reduction. Now, the last of the subset of disorder that we will look is the neurodegenerative disorder. Now, here, if we look, so we'll start with here the homeostatic microglia, and what is happening is like the microglial activation and neurodegeneration. Neurodegeneration is happening in this whole of the diagram. So what is happening is like aging. With aging, let's start with here. The aging induces microglial activation by activating the cyclic GMP and the AMP synthesis, and they are the stimulator of the interferon genes, like for example the sting signaling pathway. And what is happening is like the misfolded proteins and protein aggregates, they induce microglial activation by impairing the microglial autophagy. Now, stage 1 damaged DM, it represents a transitory and a functional subtype with a greater capacity of phagocytosis that is initiated by a TREM2 independent mechanism. And what is happening is like whereas when it goes into the further stage, that is TREM, like the stage 1 DAM goes to the stage 2 DAM. Now here this microglia, the second one, is a dysfunctional microglia. So the initial one was there in uh, the initial one was in the transitory state. It might be cleared and ultimately will be removed. But if it persists through the further mechanism like the TREM2 dependent mechanism, it might lead into this stage that is called as the stage 2 DAM. Okay. Damaged altered microglia, that is a dysfunctional microglia that is being there. Now this microglia will lead to a chronic neuroinflammation, and it leads to the activation of pro-inflammatory cytokines which damages the neurons and synapse. Further, it will lead to activation of other immune mechanism, and with these subset also, it will lead to increase in the CD8 T cells, which will lead to neurotoxicity, the neurotoxic factor, and ultimately there will be propagation of protein aggregates into the brain region. So from a homeostatic microglia, TREM2 dependent mechanism will lead to a state that is called, it's a transitory stage of stage 1 DAM. Later on, it might be either cleared, but because of the alteration, it might persist forward and it will be there in the stage 2, which is a dysfunctional state, which leads to chronic neuroinflammation and ultimately will lead to aggregation of protein in various parts of the body. So this is one of the uh, common pathway that is noted in neurodegenerative disorder. Let's see how microbiota, these subset of microorganisms play a role in the pathogenesis of the neurodegenerative disorder. And when we look into the Parkinson's, okay, so if we start with this, the first one, the short chain, the short chain fatty acids. So overall, the short chain fatty acids, they demonstrate a beneficial effect. Okay, so they are having neuroprotective effect via the G protein coupled receptor activation, and they are also having the decreased glial activation, because if you look, the subset has to be suppressed so that such kind of dysfunctional state is not reached. Now they are able to maintain barrier integrity and also are able to maintain the blood brain barrier integrity, the both the intestinal and the blood brain barrier integrity. Also, the probiotics and prebiotics are able to maintain the level of short chain fatty acids. Now, let's look at the bile acid, the ursodeoxycholic acid or the toro UDCA. They are also having the neuroprotective factor. They also reduce the glial activation and decrease neuroinflammation. Now, these also like both the short chain fatty acid and the bile acid, they reduce the glial activation and decreases neuroinflammation. Now, here is TMAO. So the first two are causing a decrease in the glial activation and maintains a state where there won't be unnecessary glial activation, and there will be a neuroprotective role both by short chain fatty acid and bile acid. But trimethylamine N oxide, this is a detrimental compound which leads to increased glial activation and neuroinflammation. So the first two are able to maintain a balance in which there won't be excessive or it decreases the glial activation. The third one, the last one, it increases the activation. Now, so we'll just see this in a while that what is happening. Okay. Now, so neuroprotective role is shown by these two and it is neurodetrimental. Now, let's look into the other side of the picture. And here the branch chain amino acid like the leucine, isoleucine and valine, they are also having the beneficial effect the in Parkinson's disease, and they also provide they promote the anti-inflammatory microglial phenotype. Now, the probiotics, be it lactobacillus, they increase the branch chain amino acid, and ultimately they maintain the barrier integrity, reduces the glial activation. Also, the tryptophan and indole derivatives, when we were discussing initially the local production of neurotransmitters and how it might impact the functioning of the brain, there we discussed regarding the metabolites of neurotransmitter which is being altered by the subset of gut microbiota. There we have discussed regarding how it might change and how it might impact the brain. So one of the mechanisms where we which was proposed was alteration in the uh, in the very basic building blocks of the neurotransmitter. So tryptophan and indole derivatives, and here also if you see the metabolism through the gut microbiota, the protein is being reduced to tryptophan and ultimately indole and its derivatives will be formed. It is also having a neuroprotective role. So if uh, the short chain fatty acid, the bile acid, branch chain amino acid, and the tryptophan indole derivative, if these are lost, okay, how these can be lost by the loss of microbiota. So all of them contributes to neuroprotective role, but since they are lost, there will be a loss of neuroprotection. And in just few slides, we'll see how this is contributing to the glial activation and ultimately damage to the brain, the TMAO. Okay. Similarly, in the Alzheimer also, the short chain fatty acid is considered to be neuroprotective in nature. Also, so the polyunsaturated fatty acid, here we see that it increases the cognitive function and are associated with the omega-3 fatty acid is associated with increased cognitive function. Omega-6 fatty acid, it leads to increased microglial activation. And the tryptophan, again, it decreases the neuroinflammation and thus is having a protective effect. Similarly, the bile acid is also causing neuroprotective effect. But again, here there is a role that is of TMAO. Okay, so the trimethylamine N oxide, again, here is one of the factor which is detrimental to the brain, both in Parkinson and also in Alzheimer. And the mechanism by which it can enter, we have already discussed, and how the short chain fatty acid production will go down, or be it the tryptophan or the bile acid, the various kind of gut microbiota. If they are lost, these neuroprotective factors will be lost. Now, uh, okay, just coming to these slides, we'll just have a look at this slide and then we'll go back. So here, if we just look, so this is an intact blood-brain barrier, and the intact blood-brain barrier is able to regulate the passage of the molecules and nutrients and the immune cells into the brain parenchyma. Now, the tight junctions, adherent junction is there in the brain. So they improve the integrity, the short chain fatty acid and other molecule are able to maintain this integrity. Now, in this disease state, what is happening is like whenever there is a disrupted blood brain barrier, it facilitates the entry of toxin, bacteria, protein, and peripheral immune cells. Now, The moment there is a disruption in the blood-brain barrier, what will happen is like the glial activation and neuroinflammation will happen because of the unrestricted access to the toxin, bacteria, and the immune cells. And also there will be an entry to the TMAO that we are discussing here that will lead to the damage to the cells. So the gut brain axis and the blood brain barrier, if you just look, the kind of integrity that is needed is somewhat having a bit of similarity. Okay, now let's look into some of the other psychotropics. We have covered antidepressants in a separate section. Here we'll look into another set of uh, like we'll start with antipsychotics. So in humans, if you look olanzapine, the microbiota, the communities of in the patients who are treated with olanzapine and the patients who are not treated are significantly separated, and these three genera are differentially abundant in the two groups. So although the cross-sectional study on psychiatric patient, no significant differences in microbiota combination composition at baseline was noted in these two subsets. In the users who were given uh, olanzapine and in those who are not given olanzapine, but the individuals who are not given olanzapine have increased in Alice types, and the patients who were treated with them, they have decreased diversity compared to the non-treated patients. Similarly, for the risperidone also, the microbiota communities have shown a significant difference between the individuals who are being treated with risperidone and also not treated with risperidone. Okay, now here also, these are all in vitro studies. I'm just highlighting on the studies in which humans are involved. Okay, in aripiprazole also, there are a significant difference in the two subsets, the one receiving aripiprazole and those who are not receiving aripiprazole. Now, we have covered antidepressants, and let's look at the lamotrigine, lithium, and valproate. So they also affect. However, these studies are there in the in vitro. Opioid analgesic, for example, methadone in human chronic opioid use in cirrhotic patient, it induces changes in microbiome composition, and specifically there is a reduction of this subset. And in morphine, also similarly, there is a reduction of the similar subset to that of methadone. In tramadol also the same Bacteroideaceae family is being reduced both in methadone, morphine, tramadol. Now coming to other drugs, like for example, if you look, so heroin, here also there is a composition and diversity of intestinal microbiota, and there is a difference between the individuals who are taking heroin and who are not on heroin. Similarly, it is going for methamphetamine. For alcohol, if we just look, human who consume alcohol, human alcoholics with dysbiosis have lower abundance of bacteroidetes and higher of proteobacteria. And alcohol dependent subjects have increased intestinal permeability, which is linked to significant microbiome alterations. In nicotine, per se, the results are there in the in vitro studies, but here in human, uh, just a minute. Yeah. So the tobacco smoker displayed a higher relative abundance of uh, 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 the subset of gut mycobacteria, that is the Prevotella and lower Bacteroides, and lower the diversity that is being used to control to compare between the controls. In xanthines, like consumption of caffeine, consumption of 3 cups of coffee daily for 3 weeks in healthy volunteers, it increases the population of bifidobacterium. However, in some subjects, there is a specific increase in metabolic activity of bifidobacterium. So these are the some of the psychotropics which is being causing a difference in gut mycobacterium. So have to be cautious while using them. Human studies I have only covered, others are in vitro studies. Now, if you look into the role in other diseases, whether there is a role of the host microbiome in other diseases or not. So yes, apart from the psychiatric illness, if you look, there is a complete lung microbiota, urinary microbiota, vaginal, skin, oral, nasal microbiota. So all of these will be involved. So uh, that is why when we compare and when we look through the pathophysiological mechanism, that is why this whole thing, first I have covered the pathophysiology and then later on I have moved to each of the disorder. So the crux is that a state of neuroinflammation or a state of low level systematic inflammation is there. So it might be possible that other subset of disorder, be it from lung, nasal microbiota, oral microbiota, or skin microbiota, might be involved when like when we see our patients who are having gut dysbiosis. So these other subset of disorders which are also having like which is also being regulated by the by the microbiota, they might be having an overlap. So the other disorder, they might be also seen in the psychiatric patient. Now, the integrative human microbiome project. So here, so like if you just look through this, so the human microbiome project was being carried out by NIH, National Institute of Health, in two phases. So this was the first one, and this is the second phase. It was a 10-year-old uh, program that organized into two phases, the HMP1 and HMP2, and they developed reference sequences, multi-omic data sets, computational and statistical tools, and analytical and clinical protocols. And they were as resources for the broader research. And the HM1, the HMP1, it basically focused on characterization of the bacterial communities from numerous body parts. So here the body parts have been mentioned, like the oral, nasal, vagina, the gut, and the skin, in a baseline study of healthy adult subjects, and included a set of demonstration projects that focused on specific diseases or disorders. Okay, the HMP2, the lower part, it expanded the repertoire of biological properties, and it analyzed both the host and the microbiome in three longitudinal cohort studies of representative microbiome that is associated with pregnancy, that is the preterm birth, which will be looking into the vaginal microbiomes of pregnant women, the inflammatory bowel disease, which will look into the gut microbiome, and pre-diabetes, that is the gut and the nasal microbiome. So these studies followed the dynamics of these conditions through multiomic analysis of multiple measurement over time, and including changes in the microbial community composition, the metabolomic, the viromics, and gene expression and protein profile from both host and microbiome and host specific properties such as the genetic, epigenetic, antibody, and cytokine profiles. And this was one of the projects which was involved in looking into the microbiome and their role. And see just to show you that so a large level study is also going on which has been done, which shows the role of gut microbiome or the other set of microbiome, be it in any part of the body which is exposed to the environment, is having a definite role in shaping the subset of disorder, be it psychiatric or be it any other subset. So,
Dr. Manish, shall we? Okay, so we
sir,
now,
like,
yeah.
yeah, yes, sir. Thank you all for listening patiently to me.
Uh, that's pretty interesting and extensive information on an emerging segment of mental health care. There are a few interesting questions too. Dr. Rajkumar, should I take them or you will take them?
No, you take them.
Right. So then Dr. Bhalbhadra Jadeja was the first to ask an interesting question here to say that can the changes in the microbe, I think there's a bit of a spelling mistake, but what he wants to say is that can we do the microbe, the GI tracks, can it be a biomarker? Can we use this as a microorganisms in the GI? Can that be used as a specific biomarker for clinical features? Are there any screening tools?
Sir, like I have covered this in the segment where we were discussing the bipolar disorder, when they are trying to develop it a kind of a biomarker for identification of the manic or the depressive episode. Um, so this is a work in progress, sir, but any screening tool as of now, sir, I'm not aware of. But like I have been through, but I was not able to find any like validated screening tool as of now. But I'll definitely look up to it and will reply back, sir. But
Dr. Jadeja, I believe your question is answered. There is a, so we, at this point in time, it is more of a theory than practical application. Right, Dr. Manish?
sir, like it is a work in progress, as you have said correctly, sir, that it is a kind of thing that is being in the process of development.
As interesting and as I was listening to your talk rather attentively, there is, what is the relationship between men and mice? I mean, are we the same as far as the the organisms are concerned?
Sir, the the problem is that the animal studies that is being done. So, sir, sir, if we look into that aspect, it will be difficult to compare a man and a mice, definitely, sir, because of the.
I was just pulling your leg. Dr. Vishal Sinha, I think your question is already answered. Do we have specific species of different gut microbiota?
Sir, I'm so sorry to interrupt, sir. Sir, I'm so sorry to interrupt. Dr. Manish, if you could stop sharing your screen because that's a little
Sorry, ma'am.
difficult.
Sorry, ma'am. Sorry, ma'am. Just a minute.
And
Sorry, ma'am.
thank you. Sorry, sir. Please continue.
Thank you, Rukshita. So, Dr. Vishal Sinha, your question that do we have specific species of different gut microbiota linked to specific disorders at present? I think Dr. Manish did mention different microbiota at different disorders. Is that right, Dr. Manish? Would you like to quickly repeat that?
Sir, like we have, like, sir, specifically, if we try to pinpoint on like 4 to 5 kind of microbiota linked to specific disorder, that will be difficult. But a family or a subspecies of a particular microbiota, if you just look, then definitely it can be broadly divided into the protective and the other being the detrimental one. So the lactobacillus bifidobacterium, these are the two subsets that we can on and like we can remember that these are protective in having a protective role as compared to the other subset. Like for example, in autism, they have shown that Clostridium is having a increased number of population in the gut. So, sir, like each of it will be having a reduction in the protective microbiota as compared to the other subset, but pinpointing one single thing, that will be a really difficult thing to answer at this point of time, sir.
Just a question that comes to me when I was reading this literature on use of uh, lactobacillus or probiotics in anxiety. It said the drug to be a probiotic, the product to be a good probiotic should have at least 3 strains of lactobacillus. Why would that be?
Okay, sir. Like the 3 strains of lactobacillus, sir.
That's
So,
right.
sir, like for example, if we just, sir, like in whatever review that I have done for that, the mostly mentioned as lactobacillus species.
Yes.
Now, if we look Actinobacter or other subset that you're talking about 3 species, so like exactly, sir, uh, it will be like what I can really think of is something related to the a whole kind of like the environment which is being created for the growth of the lactobacillus itself, which is required the other subset of species.
Thank you. There is a commercial preparation that Yakult or something, which is touted as a very popular probiotic.
Yes, sir.
Somebody tried to do a culture and found only sugar and no lactobacillus, no microorganisms in it. What is your
Sir,
experience of this Yakult?
yes, sir. When I was going through this the preparation, and when I looked at I I also looked at Yakult, sir, and there was abundant of sugar in that, and the kind of the probiotic that they have listed over there was lactobacillus casei strain that is there, and that was a single strain that they have listed over there, around 6 to 6.5 billion, sir, that has mentioned. But if you look at the product, so mostly it is sugar, that you have mentioned it correctly, sir. But the only thing that they mention is to keep it under refrigerated state below 10 degrees Celsius. So that is the prerequisite that has to be followed, sir. Like if you don't store these prebiotic or probiotic under a specified temperature, there will be a loss to the microorganisms, sir.
So -10 would be less than the
No, sir,
normal
the 10 degrees, sir, 10
10
degree.
degrees. Okay, okay. So every disorder should have some or the other GI symptoms according to this hypothesis, but it is not real. It is not seen in real world. How do we explain this?
Uh, Sir, like,
That question by Vishal Sinha.
sir, okay, sir. Sir, like, the GI upset, if we take it in that manner, definitely if you look through this aspect, what is happening is, like, sir, in depression also, the usual biological function, which includes the, like, if we take the three of the common being the appetite, sleep, and the bowel bladder movement. So the patient, they do have the bowel bladder movement alteration, which is noted in depression, also seen in bipolar uh, disorder, and also, sir, noted in ASD. Now, sir, this hypothesis, it might be a link to the same, but every patient having it might not be possible. So some of the subset definitely will have this, the GI symptom, but the other might not have. But if you look into the like, sir, the whole of the pathophysiology, that was also not only related to the GI symptom to be present. It was the whole of the total subset of population which is leading to a process of inflammation. So rather than just looking into one aspect, at this point of time, it will be better that we can broaden our view and look into the other aspects. So not only the GI symptom should be present, rather the underlying mechanism which is leading to the a state of inflammation, that should be given into consideration, sir.
Dr. Vishal Sinha is truly a clinician because he wants to now know how does this knowledge translate into management options.
Okay, sir. Like, sir, nowadays also, sir, when we look multiple these, like, although I'm not associated with any of the pharma company, but I think last week, sir, this Cognibiotic kind of thing came, and they started showing it to me. And I don't remember the exact study which they have quoted, but they are saying that the the it was containing, sir, the three strains of lactobacillus, though, sir, but when they have shown that it has shown some effect in patients with autism spectrum disorder. So, sir, like, unless and until we have a very good evidence for the same, like whether it is translating into like the occurrence of the symptoms, then only we can say that like actually whom to treat and whom not to treat for with the pre or the probiotic. So, sir, because it is mostly in theory as of now, but I hope so, sir, in coming times we'll be able to see that.
There's a related question again from Vishal Sinha. Does this also have a long-term epigenetic impact and can we utilize it clinically? I think you have answered this question. Would you like to take it again? The
Sir,
question is, does this knowledge have a long-term epigenetic impact and can we utilize it clinically?
Sir, I have one article, though I have not included in that in this presentation, which showed that because the memory part is not only there in the brain, but it is also there in the inflammatory response. So what is happening is, sir, like, whenever a bit of like a similar subset of microorganism or the similar kind of organism related to the same species, if they are having a kind of like, if it is there, the disease is there, and there is an onset of the symptom, there might be possibility of similar subset of the neuroinflammatory kind of the chain that will be activated and ultimately will lead to the symptoms. So, sir, as of now, sir, I would be happy, sir, if Bhaskar sir would add on to this part, but like, or sir, Vyang sir, if you can add on to this part, but I can share with this article to sir that they have shown to have long-term epigenetic impact with what is happening is, like, because of the neuroinflammation that is going on, there is a tendency in which the functioning of the cell that is there in the brain, it will be hampered for a long period of time, sir.
I'm going to ask Dr. Bhaskar and Dr. Malay Dave to kind of give their version, but let's just get to the questions. Dr. Pooja wants to know, could it be that patients with ASD have peculiar eating habits that lead to a disturbed gut microbiota? Which, again, chicken story, what comes first? ASD came first or the microbiota problem came first?
Okay, sir. Like, sir, the microbiota has been there since like since ever, but as far as the ASD, the diagnosis, the labeling part has come late, but the patients would have been there since the early part also. So it's very difficult to say, but ma'am, like as you have asked regarding the eating habits and the peculiar gut microbiota, so it's true. The that is why in every of the slides I have mentioned that a bidirectional kind of relationship between the two. So even if you add, if you eat the junk food or the kind of food which is not healthy per se, they'll lead to a gut microbiota. And specifically in ASD, why there is a like the answer to why per se that is not there, but they have found that there is a change in the gut microbiome for sure. But how it is happening, that needs to be answered yet.
Dr. Pooja, it's interesting that traditionally, our grandparents and great grandparents were so particular about the kind of food that a child eats, especially before the age of 4 or 5, because I guess that has something to do with the gut microbiota, but that's my guesswork. Another colleague using a Samsung SMA556E machine wants to ask is, otherwise remained anonymous, how long prebiotic must, what he means is probiotic, must be continued? He has written prebiotic, but I guess it means how long probiotic must be continued.
Okay, sir. Like as far as the probiotic is concerned, with what I know for two of the uh, molecules that are there in the market, they say it to continue for a period of 3 to 4 months.
Okay. Dr. Balbhadra Jadeja has another nice question. So, do sex hormones modulate microbiota brain axis in a way that could explain sex specific vulnerability to disorders like anxiety and depression? It's an interesting question. Is this gender variation in the prevalence of these disorders related to the gut brain axis?
Sir, there are evidence for the same, that sex hormone and the gut microbiota have a relationship between the two. And the estrogen and the testosterone, they affect the composition and the functioning of the gut microbiota. And in turn, the gut microbiota leads to the influence the level and the metabolism of these hormones. However, sir, like if you look into the effect of sex hormones which influence the gut microbiota, so they directly affect, for example, the estrogen, the progesterone can promote the growth of a certain subset of bacteria. Indirect effect, if we look, so that is also there, in which they modulate the gut microbiome growth by activating the nuclear steroid hormone receptor. So sir, there is definitely a association between the two, the sex hormone and the gut microbiota, sir.
Dr. Jadeja has another rather sharp and interesting question. From an evolutionary perspective, could modern psychiatric disorders reflect a mismatch between the ancestral microbiomes and contemporary diet, antibiotics, and lifestyle?
Okay, sir, like
That's very general question. I don't know. It only time would tell whether fast food is doing us good or harm or whatever. There is a lot of anti-fast food literature now on various groups, but I don't know whether it specifically refers to psychiatric disorders. And Dr. Jadeja would like to thank you, Dr. Manish, for very informative presentation, and he thanks you for it. Dr.
thank
Pooja
you,
has
sir.
again got a question. What could we take to improve gut microbiota except lassi, curd, and yogurt?
Sir, we have fermented food. For example, if we go and look into that fermented subset, so idli, dosa, not with much of oil. If we look, just the be it like if we look in the northern parts also, sir, in Himachal and Uttarakhand, we have this chilka roti that is made by fermented rice. So the batter is being prepared, and it's similar to what we eat in the southern part, that is idli and the dosa, sir. So that is one of the option. So all the fermented good usually we have in the condition of India is the, so the temperature, if you look, the fermentation process doesn't take that much time. So it is easy in our conditions to get a fermented food, specifically if provided with a moist environment. Usually, sir, I have seen that all the places that I have gone, so fermented food in form of rice, usually they take. So dosa is one of them, idli is one of them, and sir, chilka roti, as I have said, it's a rice batter kind of thing that is being made. So there are
However,
multiple
the
options, sir.
literature that I have read refers to
PP.
FMPP, fermented milk product with probiotics. So I don't know whether anyway, that's again a philosophical question that is beyond this topic. So Dr. Paresh Shah would want to know what is the trade name of medicines of gut microbiota useful for depression and schizophrenia? Sporolac DS or Visilec? Is it okay? What's your suggestion, Dr. Manish?
Sir, Vizilac has only a single, again, a single strain, that is Lactobacillus reuteri. Okay, sir, and similar to what you said, sir. So, like, sir, as I have mentioned, though I have no association with that company, Cognibiotic, I recently came to the to know about it. Also, sir, if we look, other subset of probiotic, usually multiple strain probiotic is to be recommended, sir. Not a single strain.
So, Dr. Farooq, that's a nice answer, Doctor. Dr. Farooq says that in an elaborate description of an emerging subject, and he congratulates Dr. Manish for this elusive presentation. But he has a question: is there an advantage on prophylactic use of probiotics? He calls it prebiotics, but is it probiotic or prebiotic? It's probiotic, isn't that right, Dr. Manish?
Sir, prebiotics are these kind of fibers which actually increase the growth of the probiotic bacteria. So
That's right.
prebiotics, like for example, artichoke or those kind of food is there, which actually increase the growth of probiotics.
So, is there any advantage in prophylactic use of prebiotics in patients with family history of psychiatric illness?
Sir, role of probiotics. So, sir,
He says prebiotics in microbiome profiling of uh, for with a family history of psychiatric illness.
okay, sir, like one of the pilot trials, sir, that I have gone through while preparing the slide, they did this in a in a subset of patients who were having they were on chemotherapy, and they did this probiotic supplementation for reducing the symptoms. And what they did was like a kind of they did a RCT, randomized control trials, but the population was around 60 to 70, and there that intervention showed that there was a decrease in the DAS-42 scores.
Okay.
Okay, so sir, this was okay, sir. This was there in last year, 2024. This pilot trial has come.
So Dr. Farooq says he wanted to know about probiotics, not prebiotics. Anyway, probably the last question that comes from Dr. Balachandra Jadeja. He says this is the last one. What is the nearest term? Sorry, what is the nearest term realistic clinical application of microbiome profiling in psychiatry? And thanks you.
Okay, sir. Microbiome profiling in psychiatry, sir. Okay, sir.
That's right.
Now, sir, microbiome profiling, as we have seen also, like there, like in schizophrenia part, sir, I have covered this, that different microbiomes is associated, microbiota is associated with different subset of symptoms. So that might be one of the application. However, sir, if you look into this, when we try to study the gut microbiota to understand the role in these symptoms, so they might contribute to the like uh, so like they might contribute to development of a different subset of symptom. So if we try to do that kind of profiling in which we are able to find a very strong correlation between one family of microbiota with a symptom, we'll be able to have a profiling done that if such and kind of bacteria or the gut mycobacteria is present, it might be possible that this individual will be having like for example, anxiety or depression, these kind of symptom. That was shown in, sir, this slide in which I covered schizophrenia, sir, where the positive, negative, and the global functioning was concerned, sir.
Thank you, Dr. Manish. I think you have covered all the questions rather very well. And like Dr. Farooq has mentioned, he thanks you and I thank you on behalf of everybody else.
Thank you,
And
sir.
may I request Dr. Bhaskar and Dr. Malay to give their inputs. We have covered all the questions.
So if I may, if I may.
Please.
Sir, can we have everybody on mute, please?
Yeah. Can this Samsung SM-M55-6B please go mute? The owner of Samsung SM-M55-6B. Please go mute. Okay.
Okay. So thank you, Dr. Manish. Quite a comprehensive presentation. I have a few points that I think are important. Point number one. What happens is that previously those areas of the body or those organ systems which we used to consider sterile are not really sterile. So the first exposure of a human being to any bacterium is in the uterus. The amniotic fluid is not sterile. And why that? Little bit of few bacterial species are necessary, because that will kind of kickstart the immune functioning of the child, so that once the child is out, he or she doesn't really succumb to a major infection and die. So first thing is that. Second thing is the gut microbiome, the future gut microbiome of an individual also depends upon the type of birth, natural birth or cesarean birth. Because the vaginal microbiome is different from the skin microbiome. So in the initial first 2 years, what is the microbiome that an individual is exposed to goes a long way in deciding what are the species that are going to be there in the gut. Third thing is that as we grow older, we have more and more number of species of bacteria, viruses, helminths. We only talk of the bacteria, that's a different thing. But we have multiple and many species which we acquire in as we grow older. So there's a developmental aspect to it. Second is the type of bacterial populations changes. So let's say as we grow older, there will be a point where we will have an increasing number of aerobic type of bacteria. And as we become older and older, that reduces and we'll have more and more of anaerobic kind of bacteria. Now, why this is important is that if you're going to do something or an intervention, something, let's say in early adulthood might not work in a later adult, because the species are changing. And it also depends on many of the factors that you said, exposure to antibiotics and illnesses and so on and so forth. So one of the most important things is the developmental aspects of how the gut microbiome develops. Second thing is that there is now an interest in two organs, which, as I was telling you in the first point, that we are looking now at and also trying to understand whether there are other systems which also have resident microbes. And two surprising ones. So see, any organ system that is exposed to the environment, so whether it's a respiratory tract, whether it is skin, whether it is GIT, whether it is the reproductive system, are exposed to the environment. So they have their own microbiome, the gut being the largest. But what is now also, in infancy is the liver microbiome and the brain microbiome. So even the brain is not sterile. So whatever we looked at in terms of neuropsychiatric disorders being associated with gut microbiome changes, maybe there is also some effect of, we have not really clearly understood what is the brain microbiome like, whether there are only bacteria there or only some products are there or something like that. But well, that is an interesting thing. And so the liver microbiome. So now these organ systems are not open to the environment. They're really closed, very well protected, but yes, they do also seem to have a microbiome. I was looking to, searching for now if it is there, does the heart have its own microbiome? So there are no studies on that. Next is, what are we going to treat? Now, if you are going to understand illnesses as collection of symptoms, we are not going to get anywhere. And so we need to look at the processes which the various microbiome populations singly or in multiples are going to affect. So the important process become reactive oxidation species, neuroinflammation, kynurenine pathways, and so on. That brings another interesting thing. I don't know whether you have read about this because I didn't see it mentioned. There is something called as the Dresden-Parkinson's model. Now, this Dresden-Parkinson model says that Parkinson's disease starts in the gut. And so one of the earliest symptoms of Parkinson's disease is chronic constipation. So what happens is the protein, it's a proteopathy. So the proteopathic changes that happen or that those are associated with Parkinson's disease actually begin in the gut. And there is a retrograde signaling because we are the vagus now going from the gut to the brain. This retrograde signaling leads to the final point where these changes settle and they spread. So starts from the gut. So somebody asked a question, are there any gut symptoms? One of this is this gut-related symptom is chronic constipation and may be associated with Parkinson's in later life. Another point is that at this point in time, I'm not going to really advise anybody any kind of probiotic therapy at all. No 3 months, 4 months, no lifelong, nothing. Because it is not one strain of anything that is involved or associated with any kind of clinical change or clinical causation of symptoms. We talk of species and we talk of strains, but then even these strains are further, they are given some numbers, 1, 2, 3.5, 4, 5, 6.7. And that's why we have multiple strains of the same genus and species in that sample. Because again, one strain is not enough. And what you have to see in a commercially marketed probiotic is the colony forming unit, CFUs. Any population which is there has to be at least 10 to the power of 7 colony forming units. Now, if these colony forming units are not there, it's a useless thing. Why we have all this lactobacillus and all? Because that is the most common listed. But that doesn't mean the other ones are not important. So lactobacillus and bifidobacterium are the commonest ones. But we have not really got our heads around the anaerobic species. Because that is one species that as we age become more and more abundant in our gut. The last thing is that there is something called as a fecal microbiota transplant, which is actually the clinical utility of the gut microbiome, not used in psychiatry as of now, used in chronic Clostridium difficile infections, chronic GI inflammations, lower GI inflammations. There have been studies and it's actually a therapeutic modality with limited success. Because if you introduce fecal microbiota transplant from healthy donors with healthy Clostridium species, you can also introduce certain kind of infections. Now, those people who are recipients have already got a compromised gut immunity. So that can create a problem. But apart from that, unless and until we have a genomic signature of the gut microbiome in a particular individual with a particular illness, if we are able to correct it, then and then only we will get clinical results in complex disorders that we deal with. So it's not going to be 1 or 2 species or 1 or 2 strains. It has got to be a complete biome genomic signature. And because the populations are varying, we have to understand that in a given individual with an ongoing illness is going to have a unique, personalized microbiome genomic signature. So if you and I have the same illness, our bacterial or microbiomic genomic signatures are going to be different. So if you're going to use a prebiotic or a probiotic, and if I use the same, we are not going to get the same results. So at this point in time, when we are talking of the gut-brain axis, we are talking as to how we are understanding this system, rather than really going all out and saying use this and this is the thing and that is the thing. We are not there yet because there are so many things, as you mentioned rightly, that the number of microbes in our body is much more than the number of cells. In fact, one of the review articles says that actually 1/3 of our body weight seems to be the microbiomes. It is that much. So just changing one species may not really cut the cheese. And we are looking only at the bacteria. We are not looking at other organisms. We are not looking at resident viruses. We are not looking at resident fungi, because that is again a huge network. We are not looking at infestations like helminths. They are there. and they're not really causing a problem, but they're doing something. So our focus is again limited. Microbiome is a big thing. We are looking only at the bacterium actually, if you want to put it that way. So that was something that I wanted to add.
Thank you, Dr. Malay. It's very useful inputs that we have put in. Dr. Manish, you started it. All the participants asked questions. Malay brought in wonderful perspective. I can see Bhaskar in now. Bhaskar, would you like to?
Yes, there are some things that needs to be said. Number one, yes, what MD has said, it's a good enough stage for what I am going to say next. Our gut microbia and we are evolutionarily linked together. Be it gut microbia, be it skin microbia, be it respiratory epithelium microbia and us, or be it urothelium microbia and us, all are related to one another via long millions and millions years of evolutionary changes, and we co-evolved. Why I am stressing here? Because the idea that microbiota is doing something may or may not be the correct idea. We need this microbia to colonize us because these areas of body are exposed to environment, and something that has exposed to environment would have microbiome in it. There will be fungi, there will be bacterium, there will be bacteriophages, there will be various virus and so on. But they are there as commensal. In ruminant, they actively help breaking cellulose. In our case, not so. In our case, they more stay as biosignature of our immune system. So for the various microbiota that we see in our various body areas, that is actually a signature of our immune system activity, our immune system composition, how and where our immune system stays in dynamic balance with these normal commensal bacteria. So if you see in that way, microbiota is nothing but an active biomarker of our immune activity. That is why giving prebiotic doesn't help, because they are just external biomarker of our immune system activity. If our immune system fails, then these microbiota, which are normally harmless, would become invasive species. And our immune system, be it mucosal immune system, be it other immune system, are just preventing that. So by understanding a person's unique microbiome composition, we can actually gauge their immune response. And that would someday be a very good biomarker of human immune system activity. So yes, microbes are important, but they are not important as something that is contributing to our well-being. That is more important as an evolutionary signature of human being, their dietary habit, their geo-environmental evolution in a certain place, and things like that. More interesting for the evolutionary biologist than for clinical application. And regarding the fecal microbiota transfer, fecal microbiota transfer has been going on for quite some time. Nobody knows how much is the placebo response in this, and
nobody knows how much is the real response in that. Because it is notoriously difficult to understand what a fecal microbiota transplant could do to a patient. The primary concern would be I should not attempt it because the immune system is already down and I should not jeopardize that. So this is my actual message for gut microbiota and gut brain axis and something. Human beings love to speculate on everything. Our diseases are not single factor. Every biological factor is present in a single dysfunction causation, and treating the microbiome would give us nothing. Microbiome is more of a biosignature of immune system function rather than anything else. That's
Thank
it.
you, Dr. Bhaskar. It's getting very, very late now. There are a couple of questions. Dr. Paresh wants to know what is this clinical application. I think this has already been discussed. And there are lots of people who have said thank you to Dr. Manish. And thank you very much for participating, Dr. Rajkumar. Thank you for having me here. And it's over to you now.
Dr. Rukshada, you have something to say?
Yes, sir. Thank you so much. Good evening, everyone. Thank you so much for joining us today. And as usual, it has been a very, very interesting and invigorating talk. A lot of us take away a lot academically and clinically as well. As the president of Bombay Psychiatric Society, I just wanted to take a moment to express my gratitude to IPS Vadodara, Dr. Rajkumar in particular, and especially a huge shout out to my friends, Dr. Malay Dave and Dr. Bhaskar Mukherjee, for this ongoing webinar series that they were they were so magnanimous about sharing their knowledge and having this open access series, and for collaborating with Bombay Psychiatric Society and giving all of us an opportunity to gain some brownie points with the rest of our psychiatric fraternity. With this, this is officially my last webinar of my tenure as president of Bombay Psychiatric Society. So just wanted to take a few moments to express once again my gratitude to everybody involved, including the delegates and participants, without whom the learnings would be lesser than what we would have taken away from this. So once again, thank you so much. And it has been an excellent collaboration and very fulfilling and very rewarding, I must say. With this, I'm going to once again thank everybody and hand it over to Dr. Rajkumar. Thank you so much.
Thank you, Dr. Rukshada. Thank you, Dr. Manish. Thank you, Dr. Vihang, Doctors Malay and Bhaskar for chipping in and making this a wonderful evening. Thank you all attendees for having sort of graced this occasion. Good night and goodbye to all of you. Meet you again in the second week of May. Till then, good night.
Thank you, sir.
Good night.